So I'm really excited to introduce our next speaker, Dr. Namrita Lall, who's joining us from the University of Pretoria in South Africa, and will be speaking about natural skincare products and some of the bioprospecting work that she does. She is a professor and research chair in medical plant sciences at the University of Pretoria in South Africa. She has been placed in the essential science indicators at the list of the top 1% of publication outputs in the field of pharmacology and toxicology. She has many accomplishments. The list is quite impressive. 132 journal articles, 24 book chapters, the UNESCO L'Oreal Award, and the Gauteng Accelerator Innovation Award from South Africa. So we are very pleased to have with us today. I should also mention, we're also very excited about this, now adjunct faculty here in the College of Agriculture, Food, and Natural Resources and the School of Natural Resources. So congratulations and welcome to Dr. Lall. Thank you.
Good morning, everyone. Very delighted to be here. Gives me a great opportunity to share, being an academic, how we are trying to translate innovation to entrepreneurship. A bit of background about the country itself. South Africa, it's a beautiful country. I mean, famous for its beautiful landscape, also famous for the game reserve, Big Five. But above all, I must say that it's very well known for its biodiversity. It's the third largest after Brazil and Indonesia. So the total number of plants found in this country is about 25,000, 1/10 of the world's population. And not much work has been done. People have not explored the medicinal potential of the country. So that's where we are working on. There are consumer products available. There are about 2050 natural health stores stocked in 2,500 pharmacies. And I would like to show a couple of them which are very popular ones. One is the red bush tea. I think some of you might know this. I saw this robust product in the supermarkets in the United States. So this is actually traditionally used for lots of medical health problems like diabetes, heart disease, cancer, treatment of colic on infants, and these are the products available in the market. Apart from that, this is another popular one, SUTHERLANDIA FRUTESCENS HERBA, which is also used medicinally for backache, edema, fever, blood purifier, and there are products available on the shelf. There's a collaborative program going on with the institution in Africa and also in Mizzou on this particular herb. It's ongoing research. So these are some, there are quite a few like this, but not much. And it means out of 25,000 plant species, only about 30 have gone into market, only have been commercialized. So that brings us, that actually encourages us to really check out the potential of the medicinal plants found in this country. If you look at the stats here, 36% of the plants are endemic. I mean, those plants which are only found in the country. So there are about 8,000 species. That is coupled with the traditional usage of the plant. I mean, people have been using these plants for various purposes for centuries. So if you take the synergy of these plant usage and the biodiversity, it makes us very exciting to see if we can really evaluate the potential of the plants and see something, if we can get something out of it. Because herbal medicine plays an integral part in South Africa. These are some of the stats showing ratio of traditional healers to the population. So this is my research area. We are trying to scientifically validate the usage of the plants the plants are selected two ways. One is based on the traditional usage, and the second one is based on phytochemistry of the plants. So now we can evaluate the potential of plants for pharmaceutical and cosmeceutical purposes, cosmetic purpose. Pharmaceutical, I would say I'm working on tuberculosis, liver protection, cancer, melanoma is my focus, but I'm working on other cancer as well, periodontal diseases, and in collaboration, usage of plants for if it can be used as a pesticide and also as herbal soap, and a whole lot of skin disorder like pigmentation, acne, wrinkle, and so on. So in today's presentation, I won't be touching on all these things. There's quite a few. So I will just be highlighting a few here and there. I will try to have a bit of technical data, not too much, and also some market survey and how we are approaching to take it to commercialization. Yeah, this is University of Pretoria. It's the largest residential university in the country, about 60,000 students, more than 60,000 students, about 1,500 faculty, and it offers about 230 degrees. There are 7 colleges, and we are based in natural and agricultural sciences. This is a picture of some of the laboratory. Infrastructure, I would say it's quite nice, but my visit here at Mizzou has been very productive thanks to my collaborators, and I see many faculties here, very approachable. So I was very impressed with the facility here. You have very good instruments, and I was very happy to visit the core facility. So there's a lot of collaboration already ongoing with Mizzou on these medicinal plants, and I foresee more collaboration after visiting the facilities in future with other faculty members as well. Okay, getting to some of the areas, I will be touching here and there a bit of scientific work, how we approached, and highlighting few things, and only talking about the success stories. I'm not talking about the failures. So when it comes to plants, first one is tuberculosis. It's quite alarming, not just in the country itself in South Africa, but worldwide. You can see here some of the stats given of incident rate. So it's the drug-resistant strain is very serious. So we are looking at, we have found some prototypes from South African plants which people can take it together with the antibiotics as adjuvant. So TB drugs are toxic to liver. And long back, 25 years back when I was a student, that time from a particular plant, the root of the plant, we found a chemical compound, very small molecule, it belongs to naphthaquinone, but that molecule was actually inhibiting 7 drug-resistant strains of TB. So at that stage, I thought I had discovered a drug, but when it went to animal trial, it was actually quite toxic. The infected animals were, it was lethal to infected TB mice, and before TB can kill the animals, this particular chemical compound killed the mice. So that was very disappointing for me. And I thought, no, let me look at the semi-pure fraction and not just look at the chemical compound. We tried to make the analogs. It's published, we got patents and this and that, but it didn't go further because of the toxicity observed in animal study. So because of that experience, I am not just taking chemical compounds further for, if we look at product development, but I'm also looking at the semi-pure fraction of the plant samples. So this is what we have got, the prototypes, which is ready to be registered as a nutritional supplement, not just for TB patient, but there are many other diseases like even cancer, where the drugs are toxic to liver. So this is the prototype developed from a medicinal plant. And then I will elaborate a bit more on the cosmetic side. That's my domain as well. Cosmeceutical, it's fairly a new term used these days. Cosmeceutical, which is kind of bridging between pharmaceuticals and cosmetics. So cosmetics which has got medicinal effect, these days people call it cosmeceuticals. Probably when we compare with cosmetics, cosmetics work on the upper surface of the top layer of the skin, whereas cosmeceuticals, it kind of penetrates in the dermis. So that is why the concentration can be less, one can use it less, and it can have, the effect can be longer than cosmetics. Now, usually it's a saying that beauty lies in the eye of the beholder. Is it really true? External beauty and internal beauty, which one is more important? I would say a combination. I mean, we all want to look good. We want others to think that we are pretty. So, moreover, the industry, cosmetic industry, it's a multi-billion-dollar industry. So, there are, every day there's innovation, and people are looking for alternatives for some or the other reason. So that is why, that brings it to the point why we are working on this cosmetic research. And here and there, I will touch a bit on some of these ailments. First of all is pigmentation disorder. Pigmentation disorder, it can be dual. It can be less pigment formation, like the white patches we see, or it can be overproduction, and that can lead to this kind of problem. So when it comes to these white patches, I just want to highlight that normally, people think of leukoderma or vitiligo kind of thing, the white patches. But in our, by reading and all, we found that there is another disease which not many people are aware of. It's called PMH, progressive macular hypomelanosis. And the symptoms are very similar to vitiligo and leukoderma. It's white patches, but when this is associated with Propionibacterium acne, then it is completely a different disease. It's PMH. So these days, the dermatologists, they are actually misdiagnosing it, and those who have got PMH, they are actually thinking it's leukoderma, and they are treating it for that. So this was something very nice, which we found in the lab, while reading, and we have got some lead plant samples, which are actually showing very good activity for this disease, but it should go through clinical studies, so I'm not saying that the prototype is ready. If it is successful, then for sure, but not yet. The other one is for hyperpigmentation, and here just to show that these pigments are present in our body, but if it is in a higher concentration, that can lead to this kind of disorder. In this case, I'm happy to share that we've got one prototype which has been commercialized by a national and international company. And this is actually available. The next one is acne. Case of acne, we all know that it's an inflammatory disease. It's due to the inflammation of the sebaceous gland. It affects about 90% of the teenagers. That's the stats. It's quite alarming. 9.4% of the global population is infected. And in this case, I'm not going through what R&D we did, but it has, for each prototype, about 10 to 12 years of R&D gone into that, and we have come up with a prototype which has got novel mode of action, and it works for this particular disease. The third one is wrinkle, anti-aging, huge market for this one. So a bit of background on this one. Our skin is exposed daily to lots of aggressive things like pollutants. But the most important, the main culprit is sun. And due to this UV radiation, it leads to the formation of free radicals, which affects the upper layer. But it's not just that. There is abnormal proteolytic degradation in the dermal layer, which is compounded with the chronic inflammation. All these things can affect the aging cascade pathway, and that leads to wrinkle formation. So what have we achieved so far? I'm not just focusing on terrestrial plants, but also the plants found in aquatic and wetland area. So in this case, we found one particular plant which is gone through all the R&D. I'm not going to go through this, but just to show you that whatever is needed in these three things, like inflammation, the enzyme activity, and few other things, we looked into that, and the results were really promising. So we took the semi-pure fraction of this sample. This is a summary of showing the main findings. How does it compare with the positive drug control? The lower the reading, the better the sample is, in a nutshell. So in this case, what we did, we took the extract, and we took the semi-pure fraction of the sample, and we added bacteria. Because these days in cosmetics, the formulation, there's sometimes people also add the probiotics. So when we think of what we eat, that actually affects our skin texture. That's very true. I mean, we do apply cosmetics, but it also, what we ingest. So these probiotics, it actually kills the harmful radicals produced in the intestine. And I was very happy to see the facility here in Discovery where the focus is on microbiome of intestine. And that could be, one can really combine the cosmetics and see what is happening in the microorganism, which is present in our body. So what we did in this particular case, we fermented the extract by adding one of the probiotic bacteria. And we were very encouraged to see that the results were very impressive. It was like 5 times better than the sample itself. So in this case, we are actually, for clinical studies, we are thinking of subjecting this one rather than the sample itself for human trial. So luckily for cosmetics, unlike pharmaceuticals, it's not ingested, it's topical application. Animal studies are not required. It's actually banned in some countries. So once we prove all the R&D and the results are convincing, not toxic, a whole lot of toxicology is done, permeation study is done, then one can subject it to human trial. But most important is, we discover something, particular plant not known for a particular disease, that is not just enough. I also look into the target. So if the commercial products are available, how do we make ours different or even better than the existing ones? Just to tell you briefly that there is a lot of enzymes involved in the aging cascade, like MMP and NF-kappa B, and a lot of cytokines, which is upregulated, downregulated. But in commercial product, we haven't found any where there is, which has got an effect on this particular protein that's called TIMP. So TIMP is down-regulated, and that also leads to wrinkles. So that's where we are now. We are looking into the effect of these lead samples on this particular protein. So the target should also be novel. Only then one can compete in the market. This particular example, which has gone through everything and the prototype is ready, has proven with significant results. I'll show you, I've got some results of clinical studies as well, just to share with you. In this particular case, we have identified the compound. In actual fact, in all the samples, even if it's semi-pure fraction, for standardization purposes, we have to know the bioactive compound. So sometimes we find bioactive compounds, most of the time, better than the fraction itself. So that I always say that it's a kind of long-term research, because if we want to take a purified compound further, it needs to be synthesized, and there is a lot more R&D required. So I say, okay, we have identified the compound. We can actually promote the semi-pure fraction, what we have found, which is slightly quicker than the pure compound, but we should not also ignore the pure compound. That is something, if the facilities are good, we should try to synthesize and take it further. So this is a very nice example where I found that the pure compound showed even better, much better activity than the sample itself. One slide to show how the clinical study is done. So even if it's cosmetics, we have to do a whole lot of toxicology. And I work very closely with people working in the industry, so that they guide me very well what data is required. So in this case, we followed the guideline, actually in all the clinical studies. So first thing is, before the efficacy test, it is actually tested just on forearm to see the toxicology, whether it's irritant or not. So this picture is showing how they monitor the irritacy effect of the sample. Once it passes through that, then the actual efficacy tests are done. Like in this case, for wrinkle, the 2 parameters like visual scan and visual face. And visual scan actually measures the patchiness and scaliness on the skin, whereas visual face scan actually measures the wrinkle formation. So these are some of the pictures. So once it's not in one go the samples make it. I mean, sometimes the concentration is low, so we have to redo. Sometimes the formulation is not correct. So yeah, it's quite a process. So then we look into the commercialized product, and we see how does this compete with the existing commercial product. So these are some of the, just an example to show that current treatment inhibitors, they are focusing either one or the other enzyme. We look into that data. So it's either focused on just antioxidant, or in this particular wrinkle one, either just as a cellular regulators, or it's used as a structural supplement, one or the other. Also, it doesn't come without any side effects, like some of these are like short-lasting activity. In some cases, inability of active ingredients to penetrate the skin. Permeation is also very important. So what have we discovered? I mean, we've got this sample, which has got kind of all in one. It has got this antioxidant, it has got, this protein inhibition activity, anti-inflammatory, non-irritant, and so on. So that's why the prototype is ready. The last topic I'm touching on is skin cancer, which is said that melanoma is my focus. Some stats about the cancer itself. So many million cases, that's 2012 stats. Prediction is it can go up to 24 million by 2030, quite alarming. When it comes to skin cancer, globally there are 2 to 3 million non-melanoma cases. The previous speaker spoke about basal cell carcinoma, but there are different types of melanoma cancer, non-melanoma and melanoma. So these are some of the stats. 1 person dies every 52 minutes from melanoma. I don't know the seriousness in this country, but I think in in Australia it's very prevalent. South Africa, because of UV radiation, is the 3rd largest incidence after Australia. So it's quite serious there. There are different types of melanoma. I mean, these are non-melanoma. The basal cell carcinoma, where it is most common type, it does not spread, but this is how it looks like, occurs in the basal cells. SCC is the 2nd most common type. Its ability to spread, forms scaly red patches. But the most dangerous one is the melanoma one, which has got a high risk of spreading to other parts of the body, develops in the melanocytes, and often found in the moles. Bob Marley, we all know, he's a musician. He's actually diagnosed with melanoma in 1977. I think people know about this. It was in the palm of the hand, soles of feet, and under the nails. Initially, it was dismissed as a soccer injury under his toenail that progressed to the lungs and brain, and sadly, he died at a very early age in 36. So we are looking still for alternative. There are drugs available, but I will show you the target which we are approaching is quite different from the existing drug. This ABCD rule, so if it's a mole, it's not dangerous, but the moment it becomes one half looks different from the other one, or the border looks notched rather than smooth, color is not uniform, or the lesion is larger than 6 millimeters, then it can be alarming, it can be cancerous. UV radiation, again, sun is the main culprit. It's the major cause of skin cancer. It's responsible for 60 to 95% of melanoma cases and 90% of non-melanoma cases. So one gets cancer, yes, treatment is one option, chemo and all those things. But how can we prevent us? Should we use sunscreen so that we can prevent? So I've got dual approach in my research, and that's what I'll be sharing with you. There's a short video to show what it can do, the UV radiation to our skin.
I won't live up to any expectations. What you see is what you get. And worse yet, I can't give an explanation for why it's gray on your side. And better yet, I'll give you an indication for why you're feeling deprived. We'll never let lack of an imagination keep us locked up inside, even when the sun has died. Somehow in the city, somewhere in the city,
Actually, this video is showing that how UV radiation can actually damage our skin, and what can we do to prevent all these things. So in our case, using this similar kind of approach, we try to see if the plant samples can have an inhibitory effect on these melanoma cancer cell line. And what we discovered that few plants from South Africa, native to South Africa, were showed quite significant activity. The pure compounds in a selectivity index value was even better than the melanoma drug. And these are some of the data here. You can see how it compares with the positive control. A lot of microscopy work done in this one. We saw that, okay, the cells are healthy if the solvent control is used. If the sample is used on healthy cells, it's actually not killing the cells, but when it is subjective, when it is, when we apply it on the cancerous cell, it has got the lethal effect. There is apoptotic bodies, and there is a degradation of cytoplasm and so on. So we looked at the current treatment, and what we found, there are different approaches, like it can be immunotherapy, it can be targeted therapy, or it can be chemotherapy. And you can see here, it's all listed here that in each therapy, it shrinks the tumor, it causes autoimmune diseases. I mean, there's some or these other side effects. The targets are different. Targeted therapy, it's specific mutation in the melanoma cells. It helps to shrink and slow the growth, but it can cause liver and kidney diseases. Same in chemotherapy, it damages the DNA of melanoma cells to stop their growth. It affects both cancerous and healthy cells. But so far, there has not been angiogenesis inhibitor which can target the spread of melanoma, and that's what we are looking into. Actually found the plant samples which can inhibit this particular one. So what is angiogenesis? It's actually in cancer cells, it's formation of blood cells. So those blood cells which feeds the tumor cells, and that can lead to the growth of melanoma cells. So we try to look into the regulatory markers which can inhibit the production of VEGF, this particular protein, which can lead to angiogenesis. So that's the approach we followed, and there are many regulatory markers. I won't go into technical details, but just to give you an overview that all these things are increased, upregulated in tumor cells. So we looked into these things for this particular sample, and we found very good activity, and that is why, these are showing some of the data in the graph, the inhibition of not just the semi-pure fraction, but also the purified compound on the inhibition of these markers. So that was quite promising. Another plant, very briefly, I will mention that this is a second plant which was also very convincing on the cell line. So both of them went through the clinical studies, but not yet for melanoma cancer, not yet in melanoma model, and that's something during this visit to Discovery and all, I found that even in zebrafish, they have this melanoma model, or definitely in animals we can try out if we are looking into long-term research and get into cancer compound. But my approach, as I said, it was slightly on cosmetic angle in this case. So we thought that, okay, let's look into the topical application of these samples, and can we get something which has got high SPF value so that one can prevent cancer. So this is what we did in clinical studies, and these samples have been proven that it can actually reduce erythema, which is caused by UVB, and that can lead to melanoma cancer. So this, for all these things, once we have got all these data, we got the prototype, which is ready. It has not been commercialized as yet. These are all the prototypes in summary. I won't go into detail. This is showing a permit summary. We cannot commercialize until we get bioprospecting permit approved by the government. So all of them, it took a few years and a lot of paperwork, but we have managed to get it, and now we have been given permission to take it to the market. This is just showing the patent summary to protect IP and so on. A value proposition, I will not go through that, but it's just listed here that main thing is that in all these things, we have proven that it's got novel mechanism of action, multiple targets, unparalleled efficacy, and so on. So basically now moving into innovation, taking it to entrepreneurship. Well, being an academic, I'm still learning business, but yeah, good support from TTO and also coming to the conferences like this helps a lot. Looked into the market survey. I won't go through what is written here. It's already, you can see that the market of melanoma, chemotherapy, it's huge everywhere. There's also good revenue for sunscreen samples. This is some of the stats showing here. Then not just for that, but for other, my area that's on acne and event tone, periodontal diseases. Some of the, all these things, some of the market revenue returns are shown here. Most important is promoting South African plant, less than 1% have been commercialized. So I was quite impressed with the plants, by talking to my collaborators with whom I'm collaborating at Mizzou about the native plants of America. I mean, some of those have not been explored, so one can consider. Financial projection, I don't want you to stretch your eyes, is just to show that, yeah, we have done market survey, all these things. The idea is myself, again, being an academic, I am taking up the challenge of moving into my own startup together with my postgraduate students. And it's a very early stage. We are looking into that. I'm trying to learn how do I get investor, how do I approach, how do I get business people with me. So it's in an early stage. Approach lots of industry. These are some of the examples just showing the people, formulators interested in all these prototypes. Not just at national level, but also a few at international level as well. Business model, this is what is required, it's done. Yeah, this is the team of our startup, but just like I said, it's in a very early stage, so we of course need to get people from legal departments and business and all those who can help us for all these prototypes. So yeah, I'm kind of concluding now that in this whole process, it's not just empowering the student, but also empowering people from the community. We're trying to make some social impact. So taking it from academia, getting into innovation, entrepreneurship, and then having some social impact, what the previous speaker was saying yesterday, the speaker who spoke about all these things. So that's kind of, we are also trying to do. If the government wants to know that if people are using these plants, what do they get out of it? So it's kind of rule, regulation that whatever royalty university gets, that certain percentage of that should go back to them. So we have got greenhouse established in the community where the plants are growing. And the idea is that if they can sell these things and generate income for themselves. So I was also glad to know that in Mizzou, you already have a huge acre of land. So if there's something comes up, it's not a problem. We can grow the plants. And in that process, we need to monitor seasonal variation and height and growth and look at the bioactive compounds, all those things. So it's actually required. So yeah, at the end, it's kind of summarizing the combination in this whole process: knowledge generation, wealth creation, job creation, and so on. All these things, different technology, we have just managed to compile it into a book. Just if you are interested, I would be happy to share. And yeah, lastly but not the least, I would like to acknowledge all my national and international collaborators, faculties with whom I'm collaborating here, for inviting me for this symposium. I'm learning a lot. It was very nice to see the facility you have. I think out of 7 core facilities, I managed to visit 5 of them. And faculties were very approachable, showed me very nicely. So I was quite impressed. Lastly, but not the least, I have to acknowledge my postgraduate students, extremely passionate. And that's how I am. Thank you very much for your attention.
Thank you very much, Dr. Lall. I was really excited and pleased when Dr. Lall accepted our invitation. I really think it's a very compelling example that very well illustrates the theme of this symposium of innovation, entrepreneurship, and the translation process or the space of translation of research. So thanks so much for your presentation. I think we have time for a couple of questions. So if there are any questions, Olga and others can run the mic. Any questions for Dr. Lall? Thank you.
Great presentation. Really enjoyed it. Thank you. What plant or family of plants are you most excited about in terms of its medicinal application?
You mean of all these, what I showed?
Just in general, in the plants that you're looking at in your lab right now, is there any plant or family of plant that is particularly interesting or that you'd like to share with us in terms of its application and what it promises for the future?
Couple of them. I would go for Mircea africana. I did list the name of the plant. And the other one is Helichrysum odoratisum. So one for anti-aging, the other one for melanoma.
I think we have a question there in the back. Thank you.
I'm curious, you have a lot of different chemistries that you're identifying. At what stage of development, when you're looking at it, do you reach out to kind of industry partners to start getting involved? Is it different for different applications, or when do you kind of engage with the different cosmetic groups or acne groups?
Actually, at the very start of the research, I like to approach the industry. And I bring them on board, and then I try to get a feeling from them, what is required by the industry. And they kind of give me like a checklist. So you've got this data, now you need to get this. So it's kind of long checklist data we have. And that's why I tell that differ from one country to another, because it's also because, linked to the regulatory requirement for a particular country. So it's very important to work hand in hand with the industry. And then we go all the way, during the process, R&D. If we get results, positive or negative, we do consult them from time to time.
Perhaps we have time for one more question, if there's any more questions for Dr. Lall.
I'll ask. I have a question. So I wonder if you ever consider or maybe use any red-listed plants, so that if you might find some compounds of that plant, and it's like being a way to conserve and propagate that plant.
Very important question again. When it comes to plant selection, I'm very careful. First of all, the industry is not interested in bark and root. It has to be sustainable. So the approach is always, most of the time, the aerial parts of the plant. Though for academic purpose, we take everything. You get very good results from the bark and root and, and also, that is one thing. The second thing is, sorry, I'm taking time.
It's okay.
The extract preparation. I mean, academic purpose, the many chemists sitting here, we would go for hexane extract, non-polar extract, or methanol, but industry won't be interested in that. So we look at both, because our interest is academia, good publication, plus my interest is also combination, basic and applied, integrating both.
Thanks for your question, Olga.
Yeah, no red data list plants. Those are kind of always rejected ones.
Well, thank you very much. Thanks for the questions and for that excellent presentation from Dr. Lall.